want to talk about some of the things I think have to change about Cryo-EM, and make it much better. So that's going to be the theme of my talk, and I think many of you are much more capable of making those changes, because I'm not an algorithm developer. I'm really just somebody who develops methods. So it'll be interesting at the end. I think I'll talk about half an hour, and then it would be really nice to open it to the floor and hear what other people think and what ideas they have for some of these changes. Okay. So here we are, where is Cryo-EM right now? And we're all extremely pleased with ourselves in this community, I think it's fair to say. We're doing really well, huge exponential growth in the number of structures that's going into the PDB every year. And you really see that growth take off, 5 years ago, just around the time of the resolution revolution. And, this is 2020 just screaming up. So we're doing extremely well. We're also very happy that we're making dramatic contributions to real world problems now, drug discovery issues. These are the two spike proteins that were reconstructed by Cryo-EM very shortly after the beginning of this pandemic. And it was really 3 weeks from these labs getting that gene to getting a structure. And a lot of that time, of course, was spent in the cloning. So it really can proceed at a pace, if you like. But where would we like to be? And my contention is we have to do all three of these. We have to be better, we have to be faster, and we have to be cheaper. And I'll explain what I mean by that. So the whole resolution revolution, which happened about 5 years ago, most of the credit goes to these amazing cameras that we have, and that's for sure true. But, the other thing that really made the difference was the microscopes, which are incredibly stable, incredibly well-behaved instruments these days, and computers. The cost of computers has come screaming down. We would not be doing many of these structures, and certainly not at the speed we're doing them, unless we had these astonishingly powerful graphics cards and CPUs. So my contention then is the resolution revolution. We got better microscopes, we got faster cameras, and we got cheaper CPUs. So all of that had to happen in order for us to be where we are. We also did a ton of automation. So everything is automated now. The collection, the processing, the analysis, the reconstruction. And these beautiful automated programs allow us to collect huge data sets in the first place and then sort them out. This was one of the early GPCRs where there were a ridiculous number of images collected, millions of particles, and then only 5% of them went to the map. And that's the power of these amazing sorting algorithms. You can also sort out many different structures on the same grid. Extreme example here is 14 different ribosome structures, but of course often we have a couple of conformational states in membrane proteins, channels, and the rest of it. So all of this amazing software allows us to sort that out. So that's good. But where would we like to be? So EM is definitely on a nice exponential rise here in the number of PDB structures, but we're quite a long way from where X-ray is. And if we look at the number of the growth of X-ray structures going into the PDB, this is where they are, and we compare it to EM, this is where we are in the total number, and this is where we are in the total number per year. So we have quite a long way to go. And the reason, of course, is that X-ray structures can be done at incredible speed now. This is the X-ray community, one of the many contributions of the X-ray community to the COVID-19 drug discovery effort, where they did fragment screening. They mounted thousands of samples, and they, collected thousands of data sets. And some of this was done very rapidly. In a few days, you can collect 1,000 data sets, and that led to a whole bunch of structures coming out. So they are extremely fast compared to EM. Where else would we like to be? We'd like to be at higher resolution. This is the most beautiful structure. We're all aware of it now. This is from Shaw's lab, and one came out of Holger's lab too. And I'm always breathtaken by this. This is a map. This is not a model. Every one of those blobs is actually in the map, and we can just see an incredible level of detail. So we would like to be generally better. This is a 1.2 angstrom map. Most of the structures in the PDB are still, in the 3 to 4 range, and that's a tough range for drug discovery. We really would like to pull that under 3 and closer to 2 in order to see things of interest in that realm. And then how long does this all take? We all know we have to collect a whole bunch of images, and then while we're collecting, we can pick particles, and we can correct motion, and we can check the CTF and correct for the CTF, and then we can do 2D classes and 3D maps. And I would say it's fair to say a lot of these can be done in around on the order of 10 hours, maybe a day of time is what we're talking about. But we'd really like to be much faster than that. I would love to see that drop to the order of 1 hour. And we certainly will have to do that in order to, come anywhere close to what the X-ray crystallography community can do. We'd also like to be cheaper. And this is an illustration. As many of you know, there are now 3 national centers where you can send your grids or your samples to collect data on these, very powerful, very expensive cryos instruments. We're one of them, based in New York. And this shows where the people who are using the center come from in the country. And what you see is a giant big gap in the middle. And I think that's very much to do with the fact that the instruments are still very expensive. We have to make them cheaper and more accessible so people can get CryoEM set up in their own labs in order to send those samples and grids to the national centers. So we'd like to speed up automated data collection, and we have done that recently. A few years ago, at the beginning of the resolution revolution, we used to collect 1 target per setup, if you like. And to set up, we have to move the stage and let it settle, let it drift around a bit. We have to do a focus and a drift check, and then we can grab an image. And we used to do it this way, which means we were limited to less than 1,200 images a day. And why I'm saying less than is that, you can't do 1,200 at 70 seconds per image. You can do at most 50 images an hour, but don't forget, you also have to, get atlases and lower mag images and fill in the cryogens every now and again, etc. So this is the maximum you could expect. And then, of course, we all realize, well, you can do image shift and you don't introduce as much beam tilt as you thought. So we could do 5 targets every time we moved the stage and did the focus and drift check. And that brings us from 1,200 to 4,000 or less than 4,000 images a day. And then, of course, you can do the same thing with bigger target areas. Here we are at about 300 images an hour, 7,000 images a day. And you can do even bigger image shift. But notice here we're dropping off, right? We're going from 7,000 here to 8,000 there. And, some of this is hard to do because the further you strip the beam out, the more hysteresis there is. So at this point, we're sort of running into the fundamental limit of how fast we can grab a 40-frame movie out of the camera. We can actually do it at 7 or 8 seconds a movie, and that would take us from 8,000 to 10,000. But we often don't see that for various reasons of latency and internet and disk writing, etc. We also we pull data from our microscopes, and this shows how many, what are we doing in practice. So this is over the period of a month, and this is, 10,000 images here. So sometimes we're getting close to that, 9,000 images a day. More likely we're at about 6,000 routinely, and sometimes worse than that. And this shows the number of images per hour that we're collecting. So, it's up around those numbers we saw before. But sometimes it's a lot worse. And the reason is that the number of images you can collect depends on the grid and the microscope geometry. So in this particular experiment here, this is what the geometry we were faced with on the grid. We collected 2 targets per hole because nice big holes, and we get 30 targets per setup. In this one, we can use a slightly different geometry, smaller holes, 1 target per hole, so we get 50 targets per setup. So this 30 and 50, this is where you're losing speed. But the other very critical thing, of course, is ice thickness. Not every hole you find has ice of the right thickness. In this case, these holes here are all too thin, and in this case, these are all too thick. And we know this because we measure ice thickness precisely for every image, and then we calibrate it back to the thickness here. So there's no point in collecting bad images. If it's too thick, it's too thick. If it's too thin, it's too thin, and you're stuck. So that means your rate of data collection goes screaming down unless the grid is covered with a very nice layer of ice. The other question it's good to understand is how many images do we actually need? And in this case, here's 10 images, and that's a 3-angstrom structure for this very nice, well-behaved protein that we have. So for a good sample, say the sample was perfect, then 10 images would get you to a 3 angstrom map, and that's 2 minutes of data collection. So the potential for doing this at very high speed is there, but of course, the linchpin in a lot of the cases is the specimen preparation. That ice thickness is not well controlled, and as we all know, the sample is not necessarily very well preserved. So many of you know we've been working on this problem for a few years. We call it the Spotted On project, and the idea was that this is the way we make grids. We blot them with big pieces of blotting paper to thin away most of the sample to get to a thin film. And in doing so, we're throwing away almost all of that precious sample that might have taken weeks to produce. And we also have a very uncontrolled environment where ice thickness is quite unpredictable. And even people who are great at making grids can make 2 grids on the same day where one will have ice thickness that's totally unsuitable and one will be very nice. And so the Spotted On project was meant to get rid of both of these problems. We use piezoelectric dispensing devices. This is a 50 picoliter drop that we dispense onto the grid. And remember, we commonly with these other vitrifying devices use 3 microliters. So this is from microliter to nanoliter to picoliter. That's thousands and thousands of times less volume that we're using. And then in order to get away from this very uncontrolled blotting process, we use self-blotting nanowire grids, which controls the process better. And so this is built into a robot. We call it Spotted On. There's a commercial version called Chameleon now. And this is the piezo device. And what it does is spray these drops onto the grid as the grid races past on its way to the ethane. And so this works. We started this project in order to automate and streamline grid making because people were struggling with making it. And indeed, when it's working nicely, you get very even layer of ice. There's plenty of real estate in this strip of ice, by the way, many, many holes there. And you can collect a whole data set. Here's a couple of examples of proof of concepts that we did. And so the ice is very even, which makes it very efficient. You can just go to the strip and you just start collecting, and you're not having to chase all over the grid trying to find the perfect ice thickness. And we all know now, of course, that there's other issues with grid preparation. We often get aggregation in ice. Particles adopt a highly preferred orientation. They won't go into the holes or not efficiently. They can be partially damaged or they can be completely denatured. So we have all these problems with sample preparation. And of course, Bob Glaser has been telling us for years, and Jacques Dubochet in his very original paper said this was going to be a problem, that there is an air-water interface and it's going to create havoc with your all of these things not only create havoc, they make things very slow. They slow everything down. If you have preferred orientation, you have to collect a lot more data. If you've got aggregation, you have to collect a lot more data in order to solve your structure. So you've probably seen this before. This is work from Alex Noble at SEMC, and he did tomograms of many, many, many different single particle projects and showed definitively that yes, your protein is indeed at the air-water interface, which is where you might expect it to be, just based on the physics of the situation. And Brownian motion are banging into that air-water interface hundreds to thousands of times a second, so there's plenty of time for them to meet that air-water interface and stick. So Spotted On does help with some of these problems. It not only can give you a nice clean layer of ice of the right thickness and make that efficient, but because the grid is racing past on its way to the ethane, the sample spends less time in a thin layer. And so it solves a few of these problems. For instance, this problem of preferred orientation. If we use Spotted On so that the sample hits the grid and then 100 milliseconds later it's in the ethane, you get rid of you get rid of some of the preferred orientation. There's still time for that sample to bang into the air-water interface, but it seems to do it, it doesn't have as much time to do it, so you alleviate these problems. And, here's a kind of a titration of that. If the spot to plunge time is on the order of a second, if it's half a second, or if it's 100 milliseconds, then we get more of these side views, 20% side views here. And that's the difference between an ugly mess of a reconstruction and a decent reconstruction that can be solved. So you really have to up those number of side views. It also can help with reduce sample denaturation. This is work from Wei-Zhen Tang's lab in Chicago. And when this pre-sequence protease project they're working on, when they blotted this on the Vitrobot, what you see is that most of the particles are denatured. You only see half the structure, the other half is just denatured. So, you know, from thousands of micrographs and millions of particles, they're just getting a very small percent, you know, less than 20% of those particles contributing to the map. So again, making everything very slow and very tedious. If we switch to Spotodon, in this case, it was the commercial chameleon, then most of the protein is now not denatured, and then they can find these different states and, you know, get the job done. And this has been the case on several other similar projects. So where would we like to be? We'd like to have perfect ice and all the same thickness, so we can really take advantage of this 100 or so target setup. We can just bang those proteins out as quickly as possible. So we'd like to go much faster. And remember, when you go much faster, you do everything cheaper, because if you double the speed of data collection, it's like one Krios turned into two Krioses. And these instruments are extremely expensive, both to buy, to install, and to keep up with. So faster really does mean cheaper in this case. And we want to be better at all of this. Maybe Chameleon will help. Graphene surfaces can help keep things away from the air-water interface. Detergents can maybe help keep away from the air-water interface. So we really need to preserve our protein better so that we can go faster, and therefore it'll be cheaper. It could get even better than that. These are the current grids we use. This is and then this is this brand new grid that Chris Russo's group has produced, where they have these very, very tiny little holes. So there's, you know, 1,000 targets in this setup as opposed to the hundreds there. So this is a new grid, and because of the hole geometry, it also helps alleviate the motion that you see at the beginning of the exposure to the electron beam. So this is both faster and better, this grid, and all of that, you know, just wraps into each other. If you get better images, then you can get your data faster. So all of them are very much interrelated. We also desperately need, I think, cheaper instruments in the field, so people can make their samples out there and then send it to the national centers, as I mentioned. Perhaps instruments like Chameleon will help do that. If people can then, you know, if you could make a perfect grid and know it was going to be good, then you could make it blind and ship it to the national centers. And even better yet, you could have a cheap screening instrument nearby where you could screen and optimize your sample and then get it sent off. So what are the remaining bottlenecks then? If we can go faster and do it better and do it cheaper, we're going to have all we're going to do is move the bottleneck to another spot, right? If we can collect data very, very rapidly, then we're going to want to change that grid every hour. If we can do it in an hour, then we have to do 24 grid changes. And so a lot of this work that's being done by hand. So this is what we do to collect a grid, right? We collect an atlas, and then we look at that atlas in some magnification, and we choose squares that are likely to give us ice of just the right thickness. So that's done right now usually by a human being. So we're going to have to automate that. This is going to have to be fully automated. Once we have those squares, then of course we look at the holes, and then this we can and do. I think almost everybody automates this process of finding and choosing the holes and figuring out whether they have the right ice thickness. And then once you have that, then you shoot a higher mag image. So all of that will have to be automated. We have to be able to change the grid over at the end of an hour and, you know, get that atlas and choose the squares and do all of that work. That's a simple algorithm in some ways, and it also probably could be aided and assisted by machine learning to a large extent. You know, we have millions and millions and millions of examples of these images, and we can learn from them what are the good ones by having done the reconstruction, and then we can feed that back into a machine learning program that will then just go and choose similar things. But it could also just be done with a straightforward image processing algorithm, I think. So we will have to remove that bottleneck. And then if we want obviously, if we're going to do this, we need to be reconstructing as we go. If we're going to change a grid every hour, we need to know we got enough data from it. So we're going to have to get to a 3D map automatically, and there's still some bottlenecks there. There are starting to be machine learning general pickers. I know Topaz, the people who did Topaz are working on that. So they are starting to be them, but I wouldn't say I would say they need more work yet. So we really need a general picker so that a human being doesn't have to look at, and optimize the picking. Then, Topaz is one of those. That's the reference for that. We need a general 2D class sorter because, if the picking isn't perfect, we always have junk classes that we want to throw out and not bother with. Although you can do that in 3D as well, but then you need a 3D class sorter. And Mike Gianfrocco's group has done quite a nice job with machine learning again, of trying to get this user-free pre-processing routines. And I'm sure others will work on that, and that machine learning again may have quite a good role to play in that. And then, of course, we need to do this all in 3D because we have to do cryo-electron tomography. So here's an example from Julia Brash, who was in our lab, and they were looking at these proteins that join together two cell membranes. This is an artificial system, and this giant thing, which looks like this, is hard enough to see in these images when it's a nice clean image, and that's all you've got. But even harder is to look at these things in situ, in a big noisy cell. I mean, here we see a nuclear pore complex, and a cytosolic ribosome and a mitochondrial ribosome perhaps, and some microtubules. But those are huge structures, and they're hard enough to see and to find and to sieve out of there. And so it's going to be an enormous challenge to find proteins in this environment, bring them out, and do the averaging that you need to get to some sort of resolution. And of course, in a crowded environment like in this, inside the nucleus, you can see nothing at all. It's just a big sea of noise. It's packed in tightly there, and it's very difficult to get things out of there. And Nico Grigoriev has an idea for finding stuff in these cluttered environments that might work, and I'm sure there'll be other algorithms coming along that help solve this problem. Okay, so that's what I wanted to say, and we can chat a little bit about it. I just wanted to tell you who we are, where we are. This is the Simons Electron Microscopy Center. We're at the New York Structural Biology Center, which is up here in Manhattan. Please come and visit us post-pandemic. It's quite a big center. We have 7 Cryoses now, and then a bunch of screening microscopes. Chameleons, things like that. We have a Helios FibSem, and we are about to purchase an Aquilos 2 FibSem. So we do both the fib milling and the high-end microscopy. A center of centers. Simons Electron Microscopy Center is the mothership that was founded in 2015. And it's at the NYSBC, and it relies on member institution support, as well as the Simons Foundation. And then we brought with us to the Simons Electron Microscopy Center NRAM, National Resource for Automated Molecular Microscopy, which was founded in 2001. And that's where we developed a lot of the automated procedures for data collection, processing, and then spotted on and making samples. And then I think that should be 2018, we started the National Center for Cryo-EM Access and Training, one of these big centers that you can send your grids to for data collection. And one of its roles is really to do a lot of training. We're struggling to do that during the pandemic, but we are making some progress. And then we have the National Center for In Situ Tomographic Ultramicroscopy, and that's just started a month or so ago, and that's another of these common funded national centers. And the role of this center is to make the thin lamella that we need, using fib milling and tools like that, so that you can do tomography on them. And then finally, we've just started in the last month a new center called the Simons Machine Learning Center, and we really want it to focus on using these machine learning methods to solve a lot of these bottlenecks and problems and get us to be better, faster, and cheaper. And of course, there's a lot of synergy between all of these centers. They're all using the same similar tools, and I think the machine learning is really going to be one of the very important ways this is all stitched together. And this is the group. We have an amazing team of highly skilled scientists and technicians. We have a bunch of really talented grad students and postdocs who are the glue that glues all this together. They have very nice biological projects to work on, and that drives the technology. And then Clint and I co-direct all